So the next talk I'm going to give is about some of these rare subtypes of GIST. You heard a little bit about them today. I'm going to get into these a little bit more and some of the work that we've done in San Diego. So just as a recap, KIT is not the only driver of GIST. So as you've heard before, there's c-KIT, there's EGFR alpha or pivot-directed factor alpha. These basically signal through several pathways, but one of these being this RAS, Raft, connector pathway. And one of the things that also regulates that is the NF1 signaling. You heard a little bit about the succinate dehydrogenase, or SDH genes, and that sort of includes mutations in the genes as well as within the carney triad patients, the changes in the methylation, meaning sort of changes in the DNA that are not actual mutations. And so just focusing a little bit for a minute on genetic testing and sort of some of the ways this can be done. So one of the questions earlier from Gary was, did we use single hotspot, sort of looking for single genes like just KIT, just PDGF, or are we doing sort of broader panels? And as time has gone on, early on we were just doing single genes, and as was most people, but as time is going on, we're doing more of these sort of comprehensive panels that are looking at large populations of genes, and basically have more ability to identify changes in the genes, whether they be we call missense mutations, where it's basically one amino acid becomes another one, whether it's copy number alterations, meaning you got this one gene and you get more of it, insertions and deletions, where something gets put into the middle of a gene or it gets pulled out of it, and so a lot of the exon 11 mutations are often deletions, and then these fusion rearrangements, which are basically where two parts of it, or two genes and different chromosomes often swap with one another, and so basically it starts off with gene A and then it ends with gene B. And interestingly, it was actually there was a fusion, BCR-ABL fusion, which is not in GIST, but it's in leukemia. Chronic myelogenous leukemia is really where imatinib, or STI-571, was actually initially being developed for, because it targets that, and so here we are now 20 years later identifying gene fusions in GIST. This is from a paper we published a couple of years ago. This was using Foundation Medicine, working with Foundation Medicine, sequencing panel at the time. This is sort of early on in their experience. This is 186 patients. I know now they've got well over 1,000 patients in their database. The point here is the way this works is that each column is a patient and each row is a gene. So this top one, not surprisingly, is KIT. The next one is EGFR alpha. But really the big picture here, 30,000 foot view, is that there's a lot of other genetic alterations in GIST, and so we need to sort of think outside the box a little bit beyond just these specific driver mutations that we've really been focused on in time. So when we looked at their database, we identified is about 70% were KIT mutant, 12% were PDGF, and then we started getting an NF1, the SDHs, the BRAFs, about 4%, these KRASs, and then sort of what we described as either triple wild type or quadruple wild type, which is sort of a little bit made up in the sense that the triple wild types we just did at the time, it was prior to the SDH testing or SDH genes being part of the Foundation Medicine panel, because it was so early on in their experience. And this paper was published a couple of years ago, so we know a lot more now. But if we look at the patients that at the time we sort of called wild type, meaning that they were wild type for KIT, PDGF, RAS, RAF, as well as NF1, and compared those to the non-wild type patients, we see a couple of things. One is that obviously they're a much rarer population. Two is that they tended, the age-wise, they were both in the mean age or up to 40s. This is actually probably a selected population in the sense that we generally expect sort of the median age, like the middle point of when people develop GIST is in their early 60s. And so this is probably a younger population, probably selected by their physicians as they're trying to identify what drugs maybe to target their tumors, and they had more advanced disease. We look at these patients, they tended to be big tumors based upon their analysis. Interestingly, these wild-type ones not infrequently have lymph node metastases, as we see with the SDH ones. And then if we break it down a little bit more, we can say, okay, well, what kinds of alterations are present in these? And so as I sort of described to you before, there's these missense ones, there's copy number changes, these insertions and deletions, and these fusions. And not surprisingly, one of those common things that we saw was missense mutations. And then we saw sort of this smattering of all these other things, but lo and behold, this was back before we knew that fusions existed in the GIST. We identified a couple of the fusions in the database. And so if we look further at these patients, several interesting things pop out. So this is basically the patients that were quadruple wild type, meaning that it was at the time when SDH testing was now part of the Foundation Medicine panel. This is prior to, these patients are all prior to SDH. So theoretically, there could be some SDH patients hiding in there. You just don't know. What you see here is a couple things. One is that a large proportion of these patients have lymph node metastases. You can see here in red, and a lot of them have metastatic disease. Metastatic disease is obviously because we're selecting for a population of patients where early on in the sequencing experience, we're sending it off for the sequencing. But the lymph node, is probably most of you are aware, is pretty rare with GIST. Most GIST do not go to lymph nodes. So the only one we generally think about are the SDH ones. But maybe that's not always the case. So it's similar to before, you sort of go across, it's tumors down or different individual genes. So if we just focus on these patients, what we found is there was actually 7 different genes that were more commonly affected in these SDH, or excuse me, in these wild type patients as opposed to the non-wild type patients. And there was several genes here, and I'm going to focus on a couple of these. First one being FGFR1, so fibroblast growth factor receptor 1 is one of the ones we identified. And it turns out we had identified 3 patients with FGFR alterations. We actually identified 2 fusions, meaning basically this FGFR gene hooked on to a gene called POC3, and then this other one called TAC1. And simultaneously we identified the ETV6 NTRK fusion in one of the patients. And so at the time, what we knew was that these FGF fusions were known to exist. The FGF1s were known to exist in glioblastoma, which is a brain tumor, the aggressive brain tumor. And these FGFR1s have been reported in thyroid cancers. Now, the NTRK fusion at the time hadn't been known to be involved in GIST, and so we first identified that this was present in GIST. I think this actually, I was presenting this actually at the same meeting when the BRAF patient was getting her operation, so it all kind of came full circle. At the time, what we knew is that these infantile fibrosarcomas, some rare breast cancers, and some salivary gland tumors existed with these NTRK fusions. And right around the same time, we got a little bit scooped. We presented it first nationally and then internationally, and then another group came out with a paper that scooped us a little bit by a couple of months, basically showing that they identified in Spain a patient with an NTRK fusion as well. And it was a very similar fusion. So in the meantime, what we did was working with Mike Heinrich and Chris Corliss at Oregon Health Sciences University, we basically looked at actually the patients that they had sequenced from the Raft data set. And they identified essentially 5 patients that were quadruple wild type from patients that they had sequenced from patients within the Raft group. And to our surprise and excitement, we actually found a couple more of these fusions within the 5 patients that they identified that they could definitively say were wild type for all the sort of known genes. So really kind of reaffirming this FGF fusion and this the NTRK fusion. And so what do we know now? So there's actually, in the last couple of years, there's been a huge amount of progress in drug development, as you know. So the NTRK genes basically have a very similar downstream signaling, and the point is basically that it's very similar, and not surprisingly. Very similar to FGF, and, KIT and PGFR alpha in terms of what things are getting signaled downstream. A lot of all these genes are all sort of converging on similar pathways, so you can almost somewhat predict that this may have been something that could have been something to think about. So at the time, there was a drug that was in clinical trials called LOXO 101. It's now called larotrectinib. This was drug was being developed by company LOXO at the time as a NTRK inhibitor. And so we looked at one of the patients, and through working with Foundation Medicine, identified one of the patients that we were sequenced and that they were being treated on trial at MD Anderson. To my knowledge, this was the first NTRK patient that was treated with GIST, that was treated with the NTRK inhibitor. So the patient had failed 5 lines of therapy. You can see here on the PET scan, pretty massive amount of disease. This is a huge tumor in his liver. He got the LOXO 101, later called larotrectinib, and 8 weeks later, you can see here a pretty impressive response. And so we reported this in collaboration with LOXO and MD Anderson back in 2016. And then basically from there, sort of the floodgates opened. LOXO identified that there was a large number of NTRK fusions in a lot of different tumors, not just GIST, but you can see GIST is one of this little subset right here. And then this was data from the trial, incredibly impressive response rates, about 75%. Objective response rates for complete responses and partial responses, which ultimately several of these were GIST, including actually 2 of the complete responses were GIST patients here, noted by the arrows. And so this ultimately led to, that trial led to the FDA approval of that in histology agnostic fashion, meaning that independent of what the disease is, meaning it doesn't matter, it's all about what the genetic alteration is. And so another good example of precision medicine where you're targeting specific genetic alteration with a specific drug and not targeting it based on the histology, meaning that it's colon cancer, lung cancer, breast cancer, etc. Likewise, the FGF fusions are another one that we identified. Again, not surprisingly, the FGF genes actually have a very similar sort of pattern, if you will, of these receptors that then downstream signal through very similar pathways. Here's RAS-RAF network again. So you can sort of see here this theme that's emerging in terms of pathways that are downstream and they're very similar. Here's just an example of kind of a lot of these receptor tyrosine kinases all going through very similar pathways. So at the time when we started this project, actually looking at the quadruple Wildtype was not the game plan. The plan was to actually look at the Foundation Medicine data and at the time say, well, this is a little bit more complicated than just a single gene. It's not just KIT and PGF, but these patients are going to have probably other genetic drivers within their tumors. And when we were looking at the data, that's when I sort of stumbled on the fact that there's such a large population of patients that were lacking these other genes. And at the time, this is just in 2015, this is what we knew about quadruple wild-type GIST. So only in a short period of 4 years, we knew very little. This was a review published. And here we are, a year later, basically identifying FGFR1 fusions, the NTRK fusions, just smattering of other genes that we identified that were a little bit more common. That these tended to be patients in their 30s to 50s, that they were more common in the stomach as opposed to in small bowel and colon, sort of consistent with the notion that these quadruple wild types can occur sort of anywhere, and then that they had both distant metastases as well as nodal metastases. Somewhere around the same time, the NIH group came out with their study basically looking at the NIH wild-type clinic and identified another patient with a fusion. It turned out to be a KIT PGFR fusion. So kind of what we're starting to add on to here is that we're starting to understand that these fusions can occur basically most commonly in the small bowel, but they can occur other places, you can see here, but rarely do we see them in the stomach. There's only one case here in the stomach, and so this is sort of compiled from our study, from the Raft one I mentioned, as well as there was a subsequent study that came out of Maria Pantaleo's group in Italy, where they identified a couple other fusions you can see here in GIST. And I was mentioning to Dr. G earlier that just in looking at some of our data recently, sort of not necessarily the driver mutations, but we've identified a couple other sort of random mutations within tumors as well. So I think it's going to become an increasingly emerging theme within GIST. And so, coming back from that talk, I wrote this paper because I sort of said, the wild type really doesn't pay respect to the fact that there is some underlying genetic reason or some underlying biological reason why these GISTs are forming. We can't just call them wild type. And so it's sort of pushed to that this be called unclassified GIST if we don't know what it is. Just because we're starting to learn, here's all the different genes and all the different pathways that we're starting to learn about within these tumors. And it's not just one gene, it's not just KIT, it's not just PDGF. There's a lot of other things that we need to start thinking about, and that potentially that some of these are targetable with drugs that might target these things. And so opening this up to the concept of precision medicine. So at this point, sort of some take-home messages here is that these wild type or unclassified GISTs tend to be more common in younger people. They tend to have metastases, sort of a little bit more frequent in the small bowel in terms of location, maybe colon being the second most common. That we would not expect a lot of these to respond to standard drugs. Maybe the PDGF should respond to one of the classic drugs, but the NTRK ones shouldn't, the FGFR ones shouldn't, the BRAF ones shouldn't, and there's other genes at play that might be modifying some of these tumors as well. And so there's still more to learn. So I'm looking for
Mutation.
Okay. Quickly, I'll just jump through this really fast. Another story that we published a couple years ago now. It's all about location, Canada, Vancouver. Okay. So we already talked about this, but the point I'm going to make here is that I'm going to talk about NF1 for a minute. So here's, we know the anatomic locations of GIST, about 55% in the stomach, and then small bowel, and then rarely in the colon. What do we know in terms of locations? Well, maybe that some of the PDGFs and the SDHs probably are more common in the stomach versus NF1 and KIT exon 9s are probably more common in the small bowel. But there's a very particular area where the small bowel goes from this duodenum here to the jejunum. And just in terms of locations, it's a very unique spot in terms of how we operate on patients and what operations they need. So at the time, what do we know about the NF1 GIST? So that basically they tend to occur in patients with neurofibromatosis or NF1. This is a hereditary syndrome. That these were thought to be NF1-associated GIST were thought to be fairly rare, about less than 2%. That there's a time, there was only one reported case of an NF1 mutant GIST in a patient that did not have neurofibromatosis, meaning that they acquired the NF1 mutation, but they didn't have the hereditary syndrome where it was passed down from their parents, or sometimes some of these patients in utero will develop a mutation that then gets perpetuated through the rest of their genome or at least part of their genome. Okay? And that these genes have been associated with NF1 and GIST. Okay? And that these were in the absence of all these other mutations that we thought about, like KIT, PDGF, BRAF, SDH. So what do we find? So sort of quickly the key findings that we find. So we looked at our database and looked at the Sloan-Kettering database, and then we sort of compared that back to our data that we looked at from Foundation Medicine. And a couple things popped out. Number one, is that if you go back to the Foundation Medicine database, the rate of NF1 mutations was just about 10%, 9.7%. In our UCSD database at the time, when we analyzed this data, where we had next generation sequencing on patients, it was almost 10%, and using the data from Sloan Kettering, working with Ron DiMatteo, it's about 6%. So it's much higher than that 1.5% that we sort of all thought. Second thing we found was that there tended to be a hot spot right here at that duodenal jejunal flexure where the small bowel sort of comes out, that there was a hot spot for GIST there that had NF1 mutations. And they often had NF1 mutations, sometimes even with KIT mutations. And so we looked at our database, and let's quickly jump through this, and here's sort of our breakdown of sort of fairly similar to what you would expect for GIST. So we looked at the patients with tumors of this duodenal jejunal flexure, and a large proportion of them had NF1 mutations. So suggesting that maybe that spot is a sort of a hot spot. And combining our data, working with Sloan Kettering, we basically identified 9 patients that had tumors exactly at that one spot. Of these patients, they were in their mid-50s, a little bit more female than male. You can see that. And a little bit more non-Hispanic than Hispanic, but almost 50-50. And part of that's probably biased by San Diego being a large Hispanic population. What we see here, most of them were localized tumors. They tended to be going very from small to very large. Most of them had a low mitotic index, and most of them had a spindle cell, or about half of them had spindle cell morphology. We broke it down even further. Here's the memorial data. So they had 7 patients with NF1 alterations. 2 of these were multifocal. 5 of these were unifocal, meaning just one tumor, and 2 of these were at the same duodenal jejunal flexure. So combining all this data, what we see here, again, the way this works is down is a gene, or excuse me, down is a patient, across is a gene. These are the tumors here based upon mitotic index, so high versus low mitotic index versus unknown mitotic index here. You can see here that a large majority of these patients had NF1 mutations, specifically at the spot, and these were patients, all these patients were tested for germline testing as well, because we wanted to make sure this wasn't something hereditary. A couple of them turned out to have a germline version. One of these was thought to probably just be a non-problematic variant, and one of them was clearly one of the patients of mine that identified actually had NF1 that nobody ever knew about, and this was his first presentation of NF1. Some of these patients also had KIT mutations. We didn't really focus on it in the paper, but it also looks like it was probably a hotspot for BRAF V600E mutations as well. And then interestingly, there's another pathway that we don't have really time to talk about, but John Chen's group published about several years ago called the Notch signaling pathway, which is sort of a stem cell embryonic pathway that was actually more commonly mutated in these as well. And in fact, you can see out of the 9 patients, 4 of them had mutations in this pathway versus of all the other patients, there was only 1 patient that had one of these pathways. So there's something there that we need to learn about. So sort of in summary, we're learning that there's probably some associations between these tumor locations and mutations beyond what we already know about grossly looking at sort of individual genes. And so there's still an evolution of things to come. But the big take home would be is that in patients that have these solitary GIST right at that specific spot, this duodenal jejunal flexure, that NF1 should be certainly evaluated in those patients because a large proportion of them are going to have NF1, whether that's just somatic, meaning it's in a tumor, or whether it was hereditary, that needs to get figured out. And so we're slicing up the pie more and more, and with that I will say thank you very much. And this was done with a lot of work and a lot of collaborators from a lot of people, so thank you.
So at this time, we have time for 1 or 2 questions.
Too good.
Jerry?
The FGFR patients, have you found a drug or anything that works with those just clinical trials? Or?
Yeah, so the FGFR patients, there are a lot of FGFR inhibitors out there now, and I think there was another new one actually FDA approved, I want to say yesterday or the day before. So there's more and more FGFR inhibitors coming out. Some of them target like the FGFR1 versus 2 versus 3 versus some combination depending upon which one they are. These individual patients, one of them that we had reported, one of them was treated with an FGFR inhibitor, but very, very late in their course, and it didn't work, but it was after failing, half a dozen therapies. In terms of our individual practice at UCSD, I haven't actually seen one as of yet, because they are probably rare, and so, but based upon the assumption of the mechanism, lots of other tumors that have FGF mutations and or fusions, there should be drugs that they should be drugged.
I was wondering, is there a correlation, or I don't have to ask you this, but in our country we have found that more and more patients are having another type of cancer besides GIST. Could be related to the mutation that they have. I mean, for example, thyroid cancer, it's very common in our GIST patients. So could be related to the rare mutation status?
Yeah, so the question is, in their country, you're Chile, right?
Mexico.
Sorry. In Mexico, they're finding that a lot of patients with GIST have other cancers. And is there a mutation that's associated with that? To our knowledge right now, the answer is no. My group actually published a paper as well in the United States using like national database data, asking the same question several years ago, and it turned out in our analysis about 17% of GIST patients in the US had a second cancer. There were certain cancers that were more common, and there's sort of patterns, some that were more common before GIST, some that were more common after GIST. They tend to be often associated with very smaller GIST, so it's probably incidental GIST. They were often getting operated on for other reasons, but there is some association. So about 1 in 6 patients or so with GIST will have a second cancer of some sort. In terms of the genetics of it, the Sun Pairing Group has published a paper a couple of years ago where they basically looked at the mutations within the tumors, and there was no specific association between any like KIT mutation and developing secondary cancer. I don't know if Dr. Chi, do you have anything to add? Or
not formally that we know of. We have seen some association, but I think from a population standpoint, obviously we've seen a lot actually associated with 9/11 New York.
Yeah.
And but not formally with other types of tumors.
Just keep in mind that if you had a cancer, you are probably being scanned more frequently than the normal population is, so they're more likely to pick up that second cancer. Doesn't mean you're more likely to get it, they're more likely to pick it up because you're already being monitored. So there's that to encounter as well.
To just add to that, I will say though, for certain things like the syndromics, FGH deficient, usually they have currently setting of a syndrome, or F1 neurofibromatosis, so there are increased risk for other cancers.
That's true.
the FDH is also very sometimes renal cell, pituitary,
Theo.
yeah, and then Theo, and then the causing sarcomas.
Surgeries would be other points.
Okay, thank you very much.
Thank you, Dr. Sicklick.